Happy New Year to everybody. Have a great 2022. And it's fantastic to be back with the webinar on emerging topics in biomolecular magnetic resonance. And we have two fantastic talks to start the webinar in 2022. And we record the talks and upload them to the ASMRBS YouTube channel. So our speakers today are Isabella Felli from the University of Florence, or CERN, and Art Parma from the University of Columbia, or Columbia University, I guess. And I'm very much looking actually forward to the two presentations. I will now hand over to Lucia Banci, which will introduce Isabella Felli. Lucia.
Thank you, Marcus. Hello to everyone. It's really a great pleasure for me to introduce my colleague Isabella Felli of CERN University of Florence. Just a few words for the very few people who do not know Isabella, because I think in this community is very, very well known. She did the PhD studies in CERN, University of Florence. Then she had some research period, postdoctoral work with Geoffrey Bodenhausen in Tallahassee and with Christian Griesinger in Frankfurt. So she has very solid NMR background, and she made and is making major contribution on different aspects, and specifically the low gamma carbon-13 direct detection spectra, and she was the first to apply this approach to liquid state that was taken from solid state and applied to liquid state. She is really the first application of low gamma direct detection. And also the other field is IDP proteins, together with Roberta Pieratelli, that they develop and sequences, approaches, and got biological relevant information. And the talk of today just combine these two aspects that are NMR experiments based on carbon-13 direct detection reveal a novel motif in IDPs. So thanks, Isabella, and now the floor is yours.
So Lucia, thanks a lot for the nice introduction. I hope I can keep to your introduction. And thanks to the organizers for giving me a chance to present some recent insights we achieved on intrinsically disordered proteins through this set of experiments based on carbon-13 direct detection that we've been working on in recent years. And by now we have assembled a large set of experiments that range from a simple 2D map to acquire a snapshot of an IDP to a set of multidimensional experiments for sequence-specific assignment, all the way to other experiments to achieve additional observables. And besides the nice technical results, what I'm fascinated about is the number of novel motifs that are sort of encoded by disorder in proteins that are emerging and that are not yet described in the protein data bank. So maybe we can provide a contribution to describing a larger part of the proteome. And of course, we should take into account the structural and dynamic properties of IDPs and their impact on NMR observables. The most well-known critical point is the reduced chemical shift dispersion deriving from conformational averaging. However, the covalent contributions are maintained. And so we should pick the nuclear spins that allow us to achieve the largest dispersion, and from going from a proton to carbon to nitrogen, actually picking 3 backbone nuclear spins in our pet protein alpha-synuclein, you can see that the chemical shift dispersion greatly increases. These are reported in the same scale in Earth. And actually, this is well known. Indeed, the starting experiments we all acquire is a 2D HSQC, but why not also combine it with the CON in which the nitrogen is correlated to the directly bound carbonyl that actually provides a nice contribution to the chemical shift dispersion, being an inter-residue correlation. So as you see in these spectra, I'll have a nice resolution, and they also show prolines that are actually only 5 in alpha-synuclein, but they turn out to be very abundant in IDPs, as also realized from the early studies on disordered systems. They are among the most abundant ones in IDPs. But let me show you a real example. Here is a CBP, one of these really complex multi-domain proteins. It has been studied for many years, as you can tell by the many globular domains that you can find if you browse the protein data bank. But these account only for half of the primary sequence, and the remaining part is actually predicted to be disordered. So we thought we would focus on 3 of these linkers that, as you see, are quite long, and they are very rich, as I mentioned, in prolines. So let's have a look at this ID4 as an example. And here are the first 2 spectra we will always acquire on an IDP. And as you see in this case, the amount of proline is really high. And so the carbon detected experiments were really key to achieve a complete sequence-specific assignment deposited in the EMRB. And already from this very simple stage, initial stage, one can realize some kind of role of these prolines that are actually not uniformly distributed. Here is the distribution of the prolines in the primary sequence of ID4, and you can tell they somehow discriminate between regions of the primary sequence that have an alpha helical propensity and probably other regions that are more elongated. So actually, prolines in some cases can become a starting point. And as you can see in IDPs, they resonate in a quite isolated region of the 2D cone spectrum. So maybe we could just use carbonyl detection to focus on prolines if amide proton detection provides information already about the rest of the non-proline amino acids. And this is easily done because you can just use a band selective pulse on 15N to acquire information on this region and only get this complementary information, as I mentioned, to what you can also get through proton detection. Of course, high resolution is important. So if you decide to focus only on this region, you can do it in about 1/5 of the time, of course, if sensitivity permits. And now let's zoom in into this region. The spectrum is reported here on the left. And it's funny to note that it actually seems like an HSQC of a normal protein, a protein of the size we were studying some time ago. For example, the size of these 2 globular domains that are flanking the ID4, these flexible linkers we're focusing on. But this is just the proline residues in this linker. And if these experiments can be acquired in a short time, we can also use this reading scheme, for example, to determine 15N relaxation rates of prolines depicted here, which of course complement the ones that one would always get on non-proline amino acids. But if you look, besides the dynamic information content, here these 15N rates are about half of the ones of non-proline amino acids, which makes sense. Prolines lack an amide proton. So it's nice to note that these lines can be so narrow in perspective of looking to more and more complex systems where a narrow line will contribute to resolution. And then still reasoning that we might use this technique only to get the complementary information to proton detection, we thought it was about time to actually try out the variants of the 3D experiments to achieve sequence-specific assignment that now, of course, provide the complementary information that make assignments more robust and also help in these proline-rich regions. Okay, now another major effect that derives from loss of a stable 3D structure is that amide protons are largely solvent exposed. And actually, exchange rates with the solvent may become quite severe, in particular if you try to approach physiological pH and temperature up to broadening beyond detection. As one can tell simply by acquiring experiments on alpha-synuclein at increasing temperatures. So as one can note here, the number of peaks in the HSQC is reduced, but we can still see maybe some interesting differential properties about exchange that is worth studying. And anyway, we would like to have information on the whole protein. And one could use, for example, one of the most sensitive to the spectra, the proton-carbon correlation experiments. But as you can see, resolving 140 peaks here is definitely not easy because overlap is quite pronounced. So you may imagine we were very happy when simply acquiring the cone spectra with increasing temperature, we proved that the quality remains actually slightly even improved at physiological pH and temperature, allowing us to recover information that is not easy to be collected otherwise. And of course, we don't want to give up the proton HSQC. The HSQC and the CON will be the first experiments that one will always want to acquire. And then why not try to make use of multiple receivers to collect them simultaneously. It's not difficult to insert an HSQC before the CON without compromises as one would achieve by acquiring them separately. And so here are the spectra one can collect. They look normal, which is good, but the important feature is that they have been acquired exactly in the same time. And to start, we decided to focus on conditions in which we can see all the peaks in the through amide proton detection. But then you can actually move to different conditions, and these experiments acquired simultaneously will actually be very useful, in particular in conditions in which the sample somehow evolves. One example that we thought we could play with is, for example, when one deals with in-cell experiments or cell lysates. And so here are the same experiments acquired on alpha-synuclein in cell lysates, again in these conditions. And here is what happens when you increase the pH. So achieving them both is important to get complementary information. And finally, I was impressed that they could give some nice results also on alpha-synuclein overexpressed in E. coli cells. Of course, we are trying on model systems, so to speak. And interestingly, also in cell, the carbonyl lineages are actually narrower than the proton ones. One possible explanation is eventually exchange, but another one are also possible local inhomogeneities deriving from the in-cell environment that will affect more protons than, of course, carbon-13s. So this tells that we can, of course, use a CON side by side to the HN to the maps to have a fingerprint of our IDP. But we can also exploit carbonyl detection to access two other simple 2D maps that will provide in the simple 2D mode information about a lot more nuclear spins respect to just a 2D HN. And so here is a 2D CACO on alpha-synuclein, where one can easily see the region of backbone correlations, but one can also observe resonances of side chains that contain a carboxylate or a carbonyl moiety. The spectrum seems the resolution is not so good, but if you zoom in, here is the glycine region in alpha-synuclein, which is quite nice. And then one can also, for example, zoom in into these side chains. Here are the resonances of the 6 aspartates that are present in alpha-synuclein. So we can achieve information also about this residue, also in IDPs, but we should assign them. And to this end, it's just sufficient to actually acquire some extra information. For example, the BCACO. And look here, if you focus on your aspartate side chain resonances, you can probably assign it yourself. Here are the correlations to bridge it to the backbone, and so to achieve the sequence-specific assignment of the side chains in an IDP. For the longer ones, one of course needs the CCCO. And as a result, here is the example of the 6 aspartate residues in alpha-synuclein. At this point, we thought we should try to have an application to see whether they can contribute some interesting information. And so we thought we would use this to have a look at what happens when calcium ions are added to alpha-synuclein. As it's known that alpha-synuclein is involved in transmission of nervous signals, and so concentration jumps in calcium ions will have an effect. And actually also taking inspiration from very nice work done on this subject through amide proton detection. However, when you add metal ions in a solution, of course, the first parts of your protein that will sense concentration jumps in calcium ions will be the solvent-exposed side chains, in particularly the negatively charged ones, such as the side chain of aspartates and glutamates. So here you can find the two insets that report the variations that we observe by increasing addition of calcium ions. And as you can see, they experience this in a different way. For example, look, here is aspartate 2 that remains in the same position, while several aspartates in the final part of the sequence experience quite large shifts. The same holds also for glutamates. And reporting these along the primary sequence, here are the chemical shift perturbation of side chain carbonyls that can be compared, for example, to the ones experienced by backbone nuclear spins that of course are a bit lower as they are a bit farther from the interaction site. Then one can of course also go ahead and acquire CON spectra to follow this process. And for example, it's possible to reveal here tyrosine that's largely perturbed. But also in this bottom part, we can see the prolines, and surprising to our surprise, also 2 out of the 5 prolines were perturbed. Kind of strange considering that prolines do not have metal binding properties. So one can of course report the chemical shift changes of carbonyl and nitrogen in the primary sequence and have a final confirmation that the final part is the most perturbed. But interestingly, the 2 prolines that we noted as the most perturbed are actually flanked by 2 aspartate residues, also intensively perturbed. So it prompted us to propose that these just tripeptides in which proline is in between 2 negatively charged amino acids could act as some kind of little local motifs that even in a disordered state, such as that of an IDP, can provide a better starting point to interact with the calcium ions in this case, or to metal ions, somehow also reducing a bit the entropic loss because of the more constrained side chain of the proline amino acid. An insight that could be achieved at least better through these carbonyl detected experiments. So another nice example in which prolines have some particular yet different role is provided by osteopontin, a protein that we worked on in collaboration with Robert. And on occasion of a visit to our lab by board, we actually, as a starting point, decided we would complete the sequence-specific assignment, and the sample was actually so nice that if you go and look in detail in this proline region, you can actually, of course, easily achieve the sequence-specific assignment reported here. But if you lower the contours, the number of cross peaks of smaller intensities will appear. And as you see, they're not easy to assign just by comparison because they are somehow oriented in strange ways. So we took the time to, on the other hand, these cross peaks are very weak. They are about 1/10 of the intensity of the others. So we put a bit more sensitivity in our 3D experiments and managed to also assign the minor forms that resulted from the cis isomer of the peptide bond in which the proline nitrogen is involved. Of course, if one can determine the cross peaks, one can also have a look at the percentage of the two conformers that are reported here. And it's interesting to note that they're not all the same. There is quite an heterogeneity. And it turns out that the ones that are higher in percentage in the cis isomers are actually prolines that have an aromatic residue as a neighbor. And of course, if we can see the peaks, we can also, for example, determine 59 relaxation rates reported here. So in the cis form, these result even a bit more rigid. So we are now working on mutants of these sort of ARO-Pro pairs stimulated by these findings to actually correlate them to the stability of this compact state of osteopontin that has been studied since a long time by the group of Robert. So another role played by proline when near an aromatic residues to stabilize more compact states in IDPs. And I would like to tell you a lot more because one example I like very much are these poly-Q helices. Poly-Qs that if preceded by leucine residues become helical, identified in the N-terminal part of the androgen receptor, as well as in one of the linkers or CBP, or actually also some charge-rich regions that all findings that I think are just giving us a glimpse on all these motifs. But I feel I would now rather like to show you how we thought to try to make some use of these experiments based on carbon detection to contribute some little information to describing this virus that is somehow keeping us company since a couple of years. This is a nice picture, I think, from the protein data bank. And so when the pandemic started, we actually had a chat with our friend and colleague Vladimir Uversky. And with him, we realized that the protein that has a lot of disorder in SARS-CoV-2 is the nucleocapsid protein reported here. So that was our starting point. and the globular domains have been characterized, the 3D structures are available. And so how about the role of flexible linkers? Actually, it's since the time of the SARS-CoV, the first kind of SARS virus, that it was realized that the flexible linkers matter, as already in 2009. However, there was no high resolution information available. So we thought we could try to provide some contribution in this field, and we decided to start from a fragment of the protein, the known RNA binding domain called NTD, but also flanked by the two disorder linkers IDR1 and IDR2, also simulated by joining forces with the COVID-19 consortium. So once the protein was expressed, we then acquired our first two experiments. In this case, it's the HNHQC shows quite nicely the well-isolated signals of the NTD. And here it shows some kind of blob from the disordered regions. And of course, you can process the spectra differently to optimize resolution rather than signal to noise and do a much better job in this region. However, it's of course interesting to also look at the CON that provides in a much more clean way information about the linkers. Somehow these two long coherence transfer delays act as a relaxation filter with respect to the globular domain. This is clean information about the flexible linkers, including the prolines. And this time, it was a time, sorry, including the prolines, yeah. So these spectra were so nice, this is CBCACON, that as I was saying, it was the time of the initial time of the pandemic. And so I thought that they could be the starting material for the practicals of the students, because of course, very difficult to teach, but these experiments are so nice that in a couple of hours practical, they could actually assign the 7 prolines in the disordered regions of the nucleocapsid protein. So let me share this with you. This is, of course, the cone plane of the 3D. And if you sort of process out the orthogonal plane, after, of course, looking at your prolines and which is the preceding amino acid, then these experiments will provide the CONP provide information about the preceding amino acid. So it's very straightforward, just from a quick look at the alpha and beta to really perform the assignment.
Isabella,
and for example,
Isabella, your time is over.
I go very quickly. So here is just a game where you can just assign them yourself by looking at the correlations in the indirect dimension. And of course, the complete assignment is a bit longer, but gives us a picture of the secondary structural propensities of the flexible linkers. One last comment, if I may, is what we are trying now to do with this assignment, because the nucleocapsid protein has attracted a lot of attention from the scientific community, as you can see here, because it's devoted to packaging the genome. And so it's interesting to have a look at what happens, which is the role of the linkers in the interaction with the RNA. And to design we up with within the NMR consortium with Andreas and Sophie, and we decided to actually look at what happens when you add a fragment of RNA, this SL4, to the NTR. And as you see here, we see sort of a decrease in intensity of the peaks, And with the increasing amounts, we actually also see liquid-liquid phase separation that has been well documented, but we did not investigate it further yet. And the interesting thing is that the linkers are the first to be perturbed. And you can, of course, compare with what happens in NTD. It's the RNA binding domain, the globular one. And it's really clear that it binds with a lower affinity and that leads to phase separation. So we thought that we could zoom in more and provide more atomic resolution information to our cone spectra.
Sorry.
And
Isabella.
as you see here, these not all the glycines are perturbed. Oh, here is a zoom in the proline region. And interestingly, also another region that contains information about leucine is perturbed. You can plot all these in the primary sequence of the protein and also plot them on a model of the NTR. And the nice thing that comes out is that we have 3 regions, 2 that are very rich in arginines, flanking the NTD domain, that could provide sort of a positive patch to recognize the negatively charged RNA backbones. But also a more remote region of leucine perturbed. To sum up, here is the known NTD binding domain with a Seaborn loop that is proved to be involved in interactions with RNA. So it makes sense that also 2 flanking regions equally rich in arginines are also involved in this interaction, but these are not the only one. Also a more remote leucine-rich one is perturbed, which tells that there is a really complex interplay of all these different domains in these complex proteins that are worth investigating more with our reading scheme. So with this, I would like to thank Roberta, with whom I shared this work, several students that helped us during the years in this set of experiments, Wolfgang and Rainer from Bruker, and also these access projects that put us on with a lot of interesting projects and interactions. If anyone is interested, is welcome to try out some carbon detection Lorentz, and not only, of course, and the IDP by NMR project that stimulated us to focus on this and get in contact with, for example, Vladimir Ruversky, with whom we started this project also on the nucleocapsid protein, together with in the frame of the COVID consortium and together with Andreas and Sophie. And so, of course, thanks also to the people I share my day with, starting from Lucia to all of us. And thank you all for your attention.
Thank you, Isabella. There are some questions. Let's start from Jim Prestegard. Jim asked about 2 questions. What is the distribution of cis versus trans conformers of prolines in IDPs? Mostly trans. So this is one question. I'll ask the 2 questions together. And have you used carbon-13 direct detection on large proteins? There are advantages similar to those suggested for nitrogen-15 by Gerhard Wagner.
Okay, thanks a lot for these questions. So I think the first one I can probably answer by having a look at our plots, if I can get it. And so can I, do you see this thing? Yeah.
Yes.
Okay, good. So in our case, but it turns out is common. The major isomer is the trans, but there are several amino acids in which the population of the cis is quite high. And so I think it's very heterogeneous, and it may be something very interesting to study. There are several other reports besides this one. For example, a very interesting protein is one involved in circadian clock regulation by Kerry Parch, or other reports by Showalter that show that it's very variable and in several cases also linked to function. And concerning the other point is how do these experiments perform on large proteins? I think the logic should change completely, because for IDPs, the most useful ones are those based on carbonyl direct detection, because of all the reasoning we have done. For the large proteins, the carbonyl CSA is a killer, and so the CON experiment will not work at all. On the other hand, if one has a largely deuterated protein, as it happens when dealing with very large proteins, one can still make use, for example, of 2D carbon-carbon maps, like carbon-carbon toxins or carbon-carbon noses. And this will be sort of one of the few experiments that can be acquired in solution on very large proteins, besides proton-nitrogen TROSY, or either proton detected or nitrogen detected, and methyl TROSY. So I think it's a true 2D carbon-carbon correlation maps. And another nice thing is that these can be easily compared, for example, with maps obtained in the solid state. So this combination between solution and solid state is interesting, because in the solid state, the CSA of carbonyls is not such a big deal, as it doesn't have any major impact on line widths, as when looking in solution at proteins with a very short rotational correlation, very long rotational correlation times.
Okay, another question, John Lang. Were the perturbation thermal metal ions reversible?
You mean the perturbation, probably this alpha-synuclein case?
Yeah.
Yes, you can, thermal, I don't really know what you mean, but
you go back in temperature, do you get the same spectra?
Yeah, okay, yeah, in this sense, thanks. It's reversible. So
the same if you treat.
You can go back and forth temperature as wished, and also calcium ions can be washed away.
You treat with the DTA, okay.
Yeah, or with the DTA, of course.
Yeah. Then
so
there is a.
I think we should delay, let's say, all the other questions to the unofficial part.
Okay. Yes.
Which will happen again, so we can discuss all these very interesting points later, maybe.
Okay,
if
good.
That's okay for both of you.
Yeah, yes, it's fine, sure.
Shall I stop sharing?
Okay.
Yeah, so thank you very much again,
Thank you,
Isabella.
Thank you, Isabella.
I would hand over to Raphael Bruschweiler to introduce our second speaker. Raphael?
Well, thank you, Marcus. Can you hear me?
Yes.
Okay, great. It's my pleasure to introduce Art Palmer as our second speaker today. Art doesn't need much of an introduction to this audience in particular, and nonetheless, I would like to mention a few highlights along his career, from which there are many to choose, actually. Art was born and raised in Cleveland, Ohio, which is not too far from where I'm right now in Columbus. He got his bachelor's degree in chemistry from Haverford College in Philadelphia, and his PhD from the University of North Carolina, Chapel Hill in 1989, with a PhD thesis on fluorescence correlation spectroscopy. Art then decided to switch fields, slowing down a little bit, but only in terms of resonance frequency, not in terms of publications, to do NMR as a postdoc at Scripps in the lab of Peter Wright, where Art and I first met. His change from optical spectroscopy to NMR has been very fortunate, for both the biomolecular animal field and for those of us who are fortunate to personally know him. In 1992, Art became an assistant professor at the Department of Biochemistry and Molecular Biophysics at Columbia University, and he rose quickly through the ranks and is currently the Robert Wood Johnson Professor of Biochemistry and Molecular Biophysics and the interim chair at his department. Art is also the NMR director at the New York Structural Biology Center. Over the years, Art has made many important contributions to biomolecular NMR, in particular for studying protein dynamics on fast and slow timescales, such as spin relaxation and CPMG experiments. Many of the NMR pulse sequences he developed are widely used in our community. Art's work is characterized by novel NMR experiments combined with mathematically rigorous analysis of the results. He's not afraid of closed mathematical expression in his work, which are often complicated ones, as they can provide a level of quantitative insight that remains still unsurpassed when doing purely numerical simulations that are nowadays, of course, possible with today's computer power. From a broader perspective, since Art entered the NMR field as a postdoc, he has become a pioneer in turning protein NMR, particularly on protein dynamics, from a semi-quantitative or even qualitative technique into a fully quantitative biophysical method, the primary method, in fact, on protein dynamics, along with rigorous statistical tests. Art has received numerous awards and recognitions, including the Laukin Prize and Nakanishi Prize from the ACS. Finally, I would like to mention that Art is a co-author of the classical textbook, Protein NMR Spectroscopy, which is a must on the bookshelf of every biomolecular NMR spectroscopist. Art's talk today is about dimerization of gadolinium-11 and new analysis approaches for spin relaxation. With this, it's all yours, Art.
Thank you, Raphael, for that overly kind introduction. I might joke that my main contribution in moving to NMR as a postdoc was how happy the optical spectroscopy community was to be rid of me. As Raphael said, I'm going to tell you two stories today that are completely different. One is an application and one is really method development. All of this work is published, so I'll just tell you the highlights and try to avoid too much detail. So the first story is about cadherins. Classical cadherins are transmembrane proteins with 5 extracellular domains. You'll see the structure on the next slide. And they're important in determining cell-cell aggregation during development, wound healing, and other biological processes. And the main biology is shown on this slide. Cells that express different subtypes of cadherins aggregate together and don't aggregate strongly or interact strongly with different subtypes of cadherins. So for example, shown in panel A, 2 cells in which fibroblasts in which you overexpress N-cadherin, in one case labeled with a green fluorophore, in the other case labeled with a red fluorophore, you can see the cells mixed together perfectly well. That's true for E-cadherin mixed with E-cadherin, cadherin 6B mixed with cadherin 6B. In contrast, in the lower panels, you can see that if you express E-cadherin in one cell type and Ed-cadherin in another cell type, then the E-cadherin cells aggregate together, cluster together, and the N-cadherin cells cluster together. So E-cadherin is expressed on epithelial cells during development. Ed-cadherin is expressed on neuronal cells during development, and the E-cadherin interactions, the N-cadherin interactions then are important in segregation of the developing epithelium from the developing neuronal tissue in an organism. So our interest has emerged from a collaboration with Larry Shapiro, a crystallographer at Columbia, and Barry Honig, a theoretician, in trying to understand the detailed molecular processes that determine the mechanism and specificity of these interactions. So here is a cartoon of one of Larry's X-ray crystal structures. As I said, there are 5 extracellular domains. There's a single transmembrane helix, and then a intracellular domain that interacts with beta-catenins and the cytoskeleton. The principal dimerization interface, the transdimer, is between extracellular domains 1, as shown in this picture, and the dimerization happens through what we call strand swapping, which is an instance of 3D domain swapping. The first beta strand of the domain disassociates from the monomer, crosses over the dimer interface, and then repacks in the corresponding position to form the final dimer. This first beta strand contains 1 or 2 conserved tryptophans, TRP2 in classical cadherins of type 1, and TRP2 and TRP4 in classical cadherins of type 2. In addition, it's now known that there's, at least in some cases, an intermediate called the X dimer, in which there's crossing at the waist between the 2 extracellular domains 1 and 2. This dimer is an intermediate that accelerates the kinetics of forming the final strand swap dimer, but doesn't affect the final affinity. It's the kinetic intermediate. So again, we wanted to understand the details of this process. Our work started now more than a decade ago when Vesko Malushev was a graduate student in the group. He worked on the extracellular domain 1 of a type 2 cadherin 8. And the final result is shown here. He was able to show by relaxation dispersion experiments that the domain existed as a closed monomer with the trip rings packed intramolecularly in equilibrium with an open monomer with the trip rings at least partially flipped out and that first beta strand partially dissociated. The rate constants are shown here on the slide that he obtained from relaxation dispersion. The open monomer has a population of about 3%, and that's then consistent with a model shown on the bottom of the slide in which 2 transiently open states interact to form the strand swap dimer, and the 3% population of the open monomer is then consistent with the measured on and off rates that Vesko obtained from ZZ exchange experiments. So this was a very nice initial result, but it was just at this moment of publication that the importance of the X dimer began to be recognized, and this construct only had the first domain. So subsequently, Ying Li joined the group as a postdoc. For those of you who were expecting Chad to be giving this talk, Ying was a graduate student with Chad and came to my lab as a postdoc. So this is as close as you're going to get to a talk from Chad Rienstra, is this slide. Ying worked on the 2-domain construct of E-cadherin. E-cadherin is a type 1 cadherin. There's only a single trip residue. And again, by a combination of relaxation dispersion and ZZ exchange experiments, she was able to work out the kinetic scheme is shown here, in which 2 monomers form the transient X dimer, which then rearrange to form the final strand swap dimer. However, in this case, we could find no evidence for the flipped out trip ring. So we, of course, don't know whether that is simply too low a population for relaxation dispersion experiments, or whether in the type 1 cadherins, the trip ring, the single trip doesn't flip out, or whether it's some consequence of having 2 domains, unlike our initial study in which we had a single domain. So we wanted to go back to the type 2 classical cadherins with the 2 trip rings and start to understand the difference between single domain and double domain constructs. At that point, Hans Kost joined the group as a postdoc and started to work on a type 2 cadherin, cadherin 11. Now we're still struggling with the 2 domain construct. So again, what I'm going to tell you today is back to the single domain construct. As I said, for type 2, the two trip rings swap across the interface. There's also a more extensive dimer interface in the type 2 cadherins compared to the type 1. And as I said, this work was recently published. Here's a close-up of the extracellular domain 1 in the strand swap configuration. You can see that the red beta strand on the leftmost monomer has crossed over the interface. Rings trip 2 and trip 4 are packed into pockets on the blue monomer on the right-hand side, and correspondingly, the blue beta strand from the rightmost dimer has crossed over, and the trip rings are packed into the pockets on the leftmost dimer. In the monomer structures, the trip rings would be packed intramolecularly in basically the same positions that they are when they're packed in the dimer. Here's an overlay just of the single domain construct with the 2 domain construct. There's good overlap between the corresponding signals, suggesting that what we learn in the single domain construct is at least partially relevant for the 2 domain constructs that we want to move on to. Now, cadherin 11 primarily populates the dimer state, and we really wanted to be able to study the monomer state. I had the simplistic idea that maybe we could use the dataless pressure system developed by Josh Wand to destabilize the dimer at moderate pressure, populate the monomer, and make investigation of the monomer dynamics easier. That simple idea turned out to be, as many of my ideas, way too simplistic, and Hans quickly found as you raise the pressure, a very large number of changes occur in the spectrum. Peaks shift, new peaks appear, and eventually peaks characteristic of the unfolded state appear. So it's a multi-state process in which new species become more populated at higher pressure. And Hans did a very detailed analysis of the pressure dependence. I just show you some pictures here. But then we obviously needed to know what are these new states that are appearing. And of course, we are interested in whether these new states had anything to do with flipped out conformations of the trip rings. So to mimic flipped out states, Hans made a series of tryptophan to alanine mutations, basically all 3 possibilities, trip 2 to alanine, trip 4 to alanine, and then both to alanine. And to make a long story short, we can correlate the changes with pressure with the changes as we make mutations. So we believe that the new peaks that are appearing are representative of, under pressure, are representative of changes that occur when the trip rings flip out at atmospheric pressure, but perhaps at populations too low for us to see in the spectra. Here's some of the evidence for this, in which as we make the mutations, you can see in various residues a more or less linear shift from the dimer state being most downfield and really a shoulder on the monomer peak, shifting towards unfolded as we introduce the trip to alanine mutations. I should point out that most of these changes, you'll see You can see pictures mapping here on the right-hand side. These changes occur in the loops surrounding the trip binding pockets. Here's another example of a shift on the left, showing, again, shift towards the unfolded state, but there are also cases shown on the right in which the chemical shift perturbations are towards some unknown conformation that is not the unfolded state. So it's clearly multiple states and a fairly complicated system. We can look at the relative populations then from the linear shifts of the different states along this trajectory of the mutations. And again, they're mapped onto the picture here. Now, a key point, we eventually realized that residue tryptophan or arginine 80 is really a key residue controlling the behavior of these loops, because this residue makes bridging hydrogen bonds basically between all three of the loops surrounding the trip residues. And that's just shown here in a cartoon. So the general picture at this point was that these loops are transiently populating more disordered states, and that the degree of disorder is coupled to the state of the putative flipping out of the trip rings. So as the trip rings flip out, the binding pockets become more disordered. Of course, at this point, we want to put some more quantitative evidence onto this picture. So Hans then collected N15 CEST, CPMG, and what we call a heroin experiment following Christian Griesinger's work. And this has all been analyzed then with a 3-state kinetic model based on work that Hans had done with Mark Rance on modeling 3-state systems in CPMG and R1 row experiments. So this just shows you a little bit of the data for residues in these loops around the binding pockets. And here's really the final picture. So on the relaxation dispersion kind of time scale, the major monomer state has the trip rings packed intramolecularly. There are transitions to one partially exposed state where the trip 2 is primarily flipped out. When that ring flips out, the disorder to order transition of the BC loop shifts towards more disordered. This would be, I think, an example of what Christian would call a population shift model. And then there's a second state in which both trip rings flip out. And again, these binding loops become more disordered. On much slower timescales, then more extensive partial unfolding happens in the domain, and we're trying to characterize that more completely. So I think that the main advance over the previous models now is the recognition that when the trip rings flip out, the binding pockets become more disordered, and that means repacking to form the dimer is a kind of coupled folding and binding in the language that Peter Wright has introduced. It also raises a hypothesis that another role of the X dimer could be to ensure that when the trip rings flipped out, there are binding partners nearby so that the strand swap dimer can form faster than these more disordered unfolded states are accessed. The unfolded states, of course, potentially could lead off to aggregation or other off-pathway intermediates. That's a hypothesis we're interested in and are trying to figure out how to test. Okay, with that, in just a couple of minutes, I'll switch to the second topic, which is really about model selection when doing the model-free analysis. Again, it's example system will be the transcription factor B zip domain of the yeast protein GCN4. We've worked on this also for a very long time. What I'll tell you about is this paper by graduate student Tim Crawley that appeared earlier this year in magnetic resonance. It is based on very nice relaxation data that Michelle Gill acquired when she was a postdoc in the group. This is data at 4 static magnetic fields, and I just show you the reduced spectral density mapping for these 4 fields. Michelle's initial analysis of this data was published a while ago in a journal whose name has disappeared from the slide. Now, the usual approach and the problem we want to solve is we have a whole set of various forms of extended model-free models going from a very simple model that just contains an order parameter and an overall tumbling time up to a model that has 2 order parameters, 2 internal correlation times, and the tumbling time. And in the usual approach, we use some kind of model selection strategy based on F statistical testing or the AIC criteria or BIC criteria to select between an optimal model in more or less an Occam's razor approach. And in the usual approach, the models are selected individually on a residue by residue basis. And here's the result of applying that sort of approach to these data. We have order parameters on a fast time scale, slow time scale. Their product is just the overall order parameter. Time scale for overall tumbling. This protein has a very disordered N-terminus, so the tumbling time was just fixed in what I'm showing you at the mean for the ordered residues. And then there's fast time scale and slow time scale or internal dynamics. And the initial question you could see is highlighted very well here for the fast order parameter or for the slow time scale. Is this variation that we see biologically interesting or not? Or is it a consequence of different models being selected for different residues in this region of the protein? Now, this problem has been recognized for a very long time. Very nice work was done by Edward Dauvergine when he was a graduate student with Paul Gooley. But there haven't been, I would say, very tractable ways of applying these kinds of insights to try to get around the model selection problem. And the basic idea that we've adopted from the statistical literature, we've not done anything original, is to apply the bootstrap. And most of you by now are probably familiar with a conventional bootstrap algorithm, for example, for estimating uncertainties in parameters. You resample your data to generate a large number of pseudo data sets, analyze the pseudo data sets, and then take their standard deviation to be the standard deviation in your original parameters. This would be a widely used alternative to doing the kind of Monte Carlo simulation that we frequently do for estimating parameters. And in fact, in the statistics literature, what we call Monte Carlo estimates of uncertainties would be called a parametric bootstrap, and then resampling the data would be called a non-parametric bootstrap. Our only problem is we need enough data so that we can resample, and luckily 4 fields is just enough data. And we decided to resample in a way that would ensure that we never had the case, for example, where we used only the data at 600 in a resampled data set. So we resample J0 as one set, J1 or J omega n as one set, J omega h as a third subset, and combine them randomly. And within each subset, we don't allow more than 2 fields to occur in a sample. That ends up giving us, for this data set, roughly 7,000 resampled data sets, and that's enough to do the analysis. Now, what's new from a conventional bootstrap is that for each set, we're going to redo the model selection process. So rather than using the optimal model to refit the resampled data, we're going to, for each of these 7,000 resampled data sets, reselect the optimal model and then average over the results, which means we'll be averaging over the different models as well as the fitted parameters at each model. Now, the averaging part is easy. What's tricky is figuring out what the uncertainties should be. And we've adopted the method developed in this paper by Efron, who's one of the founders of the whole resampling field in statistics. I'm not going to bore you with the theory because that's in the paper, but here are the results. So on this process of averaging over the models is called bootstrap aggregation in the literature, or bootstrap smoothing, or bagging as a shorthand acronym. The statisticians like acronyms as much as the NMR people do. So the right-hand side shows the original result. You can see the scatter at the fast time scale order parameter. The left-hand side is the smoothed result. And now you can see that scatter has gone away. That's really the model selection error. We have a much smoother profile, and therefore we think a much better ability to discern what's biologically interesting. And we know, for example, these little bumps, or we know from MD simulations or think from MD simulations that these little bumps reflect transient formation of transient helical turns. And these turn out to be the initial interaction sites of the protein with the DNA double helix in the process of binding. Similarly, you can see that the timescales as well are substantially smoothed out in this process. So where now the fast time scale motion is quite uniform, and the slow time scale in this disordered binding region now is quite uniform at just over a nanosecond. We can then look in detail, of course, at the distributions of the resulting order parameters and timescales. And you can see here's just one interesting example. The vertical line is the result from the conventional analysis. And that's basically the sum then of the distribution, or the average of the distribution from the bootstrap sampling. But you can clearly see in the sampling that there's a bimodal distribution of two different timescales that's not caught in the conventional analysis. Again, we can look at particular details. I think I'll skip that. So the short answer is, I think this is assuming one can get data at 4 fields for doing the model-free analysis. This is a very convenient way of solving the model selection problem that Gooley and Dauvergine had really highlighted so long ago. Outside of model-free relaxation analysis, of course, there's lots of places in NMR spectroscopy and in biophysics in general in which one has to do model selection. So I think this is a general procedure that is likely to have other applications in areas that we're interested in as a community. So I'll stop there. The work I told you about today was really done by the first part by Hans Koss and the second part by Tim Crawley. And the Cadheron work is a collaboration with Larry Shapiro and Barry Honig, as I said. And of course, our ability to acquire data at 4 magnetic fields is due to the New York Structural Biology Center. So we thank the Center for Access. And thank you again, Marcus, for the short invitation.
Okay, well, thank you, Art, for a wonderful talk. We have time for some questions, and there is one in the Q&A. window by Jim Prestergaard. I don't know whether you can see it, Art, but I will read it for you. Catharians are probably glycosylated. Is anything known about how glycosylation affects these interactions?
Not by me. There aren't glycosylation sites near the dimer interface in the strand swap dimer. That's all I can really say about it.
Okay.
There's also a question from Walter Chassin.
Yeah, there is another question for Art.
Yeah, Walter Chassin writes, does the discovery of the bimodal distribution in the resample data set imply there is more to discover, or is this the result of the analysis?
Well, you could view things, in two ways for that, seeing that distribution. One way would be to say, there are two processes on two different timescales, and we don't yet know what those two different, dynamic transitions would look like. You could say it's an artifact of the resampling. The only way to test that would be then to have a fifth field to cross-validate, and we're in the process of trying to get a fifth data set. We do know that, there's plenty of residues in the protein in which we just see a single distribution that's completely centered on the results we get from the conventional analysis, with uncertainties that completely match what we would get from Monte Carlo simulations. So it's not, a totally random analysis process, but we've had to use all the data we have to do this bootstrap aggregation. So to try to cross-validate needs another field. I should say, the real interest in these different timescales, of course, is which timescales are associated with the eventual zipping up of the recognition region to form the final helical structure in the complex with DNA, and which timescales might be associated with a search in three-dimensional space for the DNA. So on the more biological side, I think those are the two principal questions that still are open.
Just as a brief follow-up, Art, you get these more uniform correlation time distributions, and correlation times in these relaxation experiments are often kind of underutilized. How well does this agree with your MD simulations that you mentioned?
Right. Still, I would say not fantastic agreement, and certainly the the agreement between MD simulations and the order parameters, these days, quite good. It should be then that the processes that have a correlation time, internal correlation time, in the 25 to 50 picoseconds, we ought to be able to start to get those well from simulation. So I think that's the next challenge. The 1 nanosecond process, right, that's still going to be tough. One really probably requires at least microsecond simulations or longer to get to those. But I think you could say from your work and many other people's work that maybe the battle between MD simulations and order parameters is almost won, and now it's time to move on to the next challenge of the timescales.
Okay. Marcus, you raised your hand.
Yeah, thank you. So, I mean, you called. I mean, you had a label of smoothing there at the bootstrapping aggregation. So, I mean, of course, that is a little bit based on the assumption that the more scattered plot is an artifact of the model selection, and that the one or the other model was badly selected, I guess. So I mean, but of course, it could be that. I mean, what's your opinion? I mean, it could also be that different models apply to different, let's say, residues. And by doing the bootstrapping and kind of averaging over different models, you kind of smooth that out and you lose that information. Or is that wrong?
Right. So I mean, the idea goes back, and suppose we acquired 7,000 data sets, which we're not going to do. But if we did, and did the conventional analysis on those 7,000 data sets, you wouldn't expect to select the same model for a given residue 7,000 times, because just because of the experimental variation from data set to data set, occasionally the minimum AIC or minimum F statistic is going to select a different model. And once you select a different model, you're going to get different parameters. So the problem is real. And of course, the difficulty is in trying to distinguish between the cases where different model selections, residue to residue, is just an artifact of the particular data set you happen to have sampled versus being a real difference in chemistry and biology. And, you know, I think we do know in cases where we do have independent data sets on different protein preps of the same protein, that if we go through the analysis and end up with the same model for a given residue, we get virtually identical order parameters, correlation times, etc. But if we ended up with a different model, then all bets are off. So we do have some evidence suggesting that the problem is real, and there's no way to acquire 7,000 data sets. So this is, you know, the bootstrap aggregation is a way of trying to take that into account with the data that you have. It does depend on the assumption that the variation in our 4 data sets, that the variation in the 4 data sets somehow, you know, is a reasonable sampling of the variation we would have seen if we had acquired 7,000 data sets. And that's obviously, you know, quite a stringent assumption. And it does tend to fall apart if we only had 3 data sets. If we throw one of these data sets out, then in fact you can't fit anymore the more complicated models. There's just not enough information in the data. So I say 4 data sets barely gets us there.
Thank you.
There's another question in the chat from Christina Thiele about residue by residue versus global fit. She asked, I was wondering whether you do not only a residue by residue fit, but also global fit. Would that be useful?
So for this particular data set, well, for us, residue by residue versus global really has to do with the overall tumbling. So in some analysis, sometimes we do the analysis in which each residue gets its own local overall tumbling time, and then we determine the overall rotational diffusion tensor from the methods that Raphael developed. In other cases, we, as part of the fitting, do assume overall diffusion tensor and then optimize the axial ratio and tumbling times of that diffusion tensor. But in all cases, the internal order parameters and correlation times are fit individually, residue by residue. The only global parameters have to do with the overall tumbling of the molecule.
Okay, thank you. There are two questions from the panel. Oscar and Christian, who wants to go first?
Thank you, Christian. So, Art, when normally use the F-test for model selection, one of the problems you found, at least, I think this is a problem, at least for me, is the accuracy in the determination of the experimental error. Is this bootstrap, like the original error bars, take into account in the calculation of the chi-square, actually, in the end, have a translation into the accuracy that you will obtain in the F-test selection? How does this address in the bootstrap? Is there any benefit on that, or is just this is the same?
Well, the problem with the original experimental uncertainties arises whether you do F statistics or AIC or BIC, or anything. So obviously, the results from any statistical analysis, are only really as good as the uncertainties in the data that you collected. Now, in this data set, I mean, the big problem can come in when you have some data point that just for blind luck has a much smaller uncertainty than reality. So in this particular data set, what we did was smooth the error bars over the 4 replicates so that there was, for a given residue, one uncertainty with the 4 J0 values that came out of the spectral density mapping. That was then the sum average of the uncertainties in each individual experiment. And that helped reduce some of the wilder fluctuations, but certainly, that's an issue. The more, the relaxation rate constants, of course, came from some relaxation experiment, and the better that experiment is, the better the uncertainties are, the better the ultimate result will be. So,
oh, that's clear.
The more duplicates one can take, for example, at the start, the better.
That's for sure.
This isn't magic. I would say this isn't the magical solution to all problems.
Sure.
Christian?
I also had a question to this last example with the backing. You mentioned that there were these, there's a helix when the autoparameters were a little bit higher. And
yeah, so I should point, if I had more time, I would belabor this. So this structure is the X-ray structure of the complex. And these blue helices in solution are the disordered parts. This part is formed all the time, this coiled coil. So these are disordered, partially disordered in solution, and you see the decline in the order parameters in the data.
Yeah.
In the MD simulations, the locations where those bumps in the order parameters are correspond to formation of transient helical turns at those locations, and those particular residues map right into the middle of the binding interface. So the simplest, or our hypothesis is that you preform the initial turn of helix that then can make the initial interaction with the DNA, and then you zip up the rest of the helix to make the final conformation. And then the other aspect, as I said, is whether these more flexible regions can swing around in space in what people call fly casting to try to find the DNA in the first place.
My question was, I mean, if one has a transient helix, and I mean, I saw that there were order parameters which were almost flat for something like 5 residues, which would make a helix, while in the backed analysis, then it was more like a well, inverted parabola or something like that. And which of the two would fit better to a I mean, a helix forming, because at least you need one winding, and that would then mean at least 4 to 5 residues or so. I mean, that was my question. I mean,
Right.
in principle, in the MD simulation or so, you might be able to see that.
Yeah, so the simulations that we did in this paper here by Paul Robustelli, there were 4 100 nanosecond simulations. It's extremely difficult to try to equilibrate the system because you have to pull it off of the DNA and somehow unravel the helices. So I think to answer a question at that detail, we would really have to go back and really try to equilibrate many more replicas and run much more extensive simulations with more modern force fields and whatever. I can't see the year here. So I think you're asking the level of detail we can't get from our current simulations.
Okay. No, just because you were saying, okay, can we now relate this with biological function?
Yes.
And that's why I was asking whether maybe you have something that relates or that explains the biology better with the backed interpretation. Okay.
I might comment there that maybe RDCs actually might be an independent NMR parameter to cross-validate which of the representations is better, especially for these nascent helices.
Right. So when Michelle Gill was in the lab, she set out to do that. At the time, we were pretty nervous about putting alignment media into this system for fear we'd be perturbing this, very delicate balance of transient helical formation, et cetera. So she tried to attach lanthanide tags to the C terminus here, far from the basic region in order to align. And all we ever did was blow up, by putting the tags there, all we ever did was blow apart the dimer. So yeah, it's a great idea. It's, we should go back and try to make that work.
Okay,
And I have a naive
question. Go ahead, Isabella.
Naive question probably, but how about other rates? Maybe some interesting carbon-13 relaxation rates?
Absolutely. There's because this is, the region that's ordered is a helix. All of the NHs point more or less in the same direction in the coiled coil, and that makes determining the overall diffusion tensor difficult, and that makes doing the rest of the analysis difficult. So carbon-carbon relaxation would be very helpful. We just haven't done it yet. So I should say there's no sign of any chemical exchange timescale processes. So it seems the N-terminal region seems sufficiently disordered that chemical shifts are averaged on a very fast timescale.
Also, do you think, Art, that the anisotropy is enough to measure the field dependence on the J coupling, to see residual bar coupling there? As Advax did. I mean, I know it's very tricky, like you need amplitude modulation measurements because this is very minimum.
That experiment sounds difficult enough that you should come back for a sabbatical and do it.
Sure.
Although I suppose one could imagine extending these extending the helices and making them longer. There's no particular reason. I mean, that's the length in the protein, but for aligning it, there's no reason not to make them longer.
So with that, I think we at least close the official